Samar Island is the third largest island in the Philippines and has one of the most extensive forest over limestone ecosystems in the country. This ecosystem is home to a wide array of uniquely adapted flora and fauna. Samar Island is known to be one of the centers of botanical diversity in the Philippines and has been nominated as a UNESCO World Natural Heritage Site for its exceptional natural beauty and precious biodiversity. For these reasons, CONserve-KAIGANGAN, a biodiversity research program, was established. Kaigangan is a Samar local word that means forest over limestone. Our research program's mission is to assess and conserve the biodiversity in Samar Island's forest over limestone towards its sustainable management. One of our program's projects is the assessment of biodiversity and community structure in forest over limestone ecosystems. This component of our research aims to determine plant diversity and vegetation structure in various habitat types in forests over limestone, to determine animal diversity associated with forest over limestone, to identify functional classification of vegetation with its associated animal diversity, and to analyze the environmental factors influencing the prevailing structure of the biota. We conducted a comprehensive biodiversity assessment of forests over limestone in the municipalities of Paranas, Taft, and Giwan with remarkable findings. A combined total of 71 plant species were recorded from these municipalities, with at least 3 tree species classified as vulnerable based on the DENR updated national list of threatened Philippine plants. A total of 52 terrestrial vertebrate animal species were also recorded, including 5 amphibian species, 15 reptile species, 20 bird species, and 12 mammal species, with conservation statuses ranging from threatened to endangered and critically endangered, based on the DENR updated national list of threatened Philippine fauna. This knowledge will empower our stakeholders in these areas to make appropriate research-based conservation efforts for the protection of these ecosystems that harbor an assemblage of rare, endemic, and economically important flora and fauna. Through this program, people will be equipped with knowledge on forests over limestone biodiversity, including the ecosystem services that it can offer to the community. We at CONserve-KAIGANGAN continue to further raise awareness and capacitate national and local government agencies, especially local communities living in and around Samar's Kaigangan.
Hello everyone, welcome to Kaigangan Biodiversity Webinar Series. In March 2022, Ms. Elaine presented about the biodiversity of forests over limestone in Samar Island Natural Park, or also known as the SINP. Her study aimed to provide information on the outcomes of the plant and animal biodiversity in SINP. Second, to describe the tree community structure in SINP. Third, to highlight the significance of the nomination of SINP as a UNESCO Natural Heritage Site. This was conducted specifically in Paranas and Taf, Eastern Samar, a province in the Philippines located in the Eastern Visayas region. Some of the highlight of the results of her study are the identification of some ecologically important amphibians, for example, the Philippine endemic Platymantis guentheri. In terms of reptiles, a very interesting one example is the Lycodon dumerilii, which exhibits a widespread distribution and has been documented throughout the Mindanao Paig or the Pleistocene Aggregate Island Complex. Ms. Elaine discusses also about her interesting findings on birds, for example, the Bucerus hydrocorax semigaleatus, also known as the Philippine hornbill, a large species of hornbill endemic to the Philippines. In terms of flora, she found this new island record species, for example, the Tectaria calcarea, a fern or herb species with native range in the Philippines. Overall, Ms. Elaine emphasized that SINP is indeed worthy of its nomination as a UNESCO Natural Heritage Site because of its natural and cultural significance.
Yeah, good morning, everyone. Welcome to our fourth episode of our Kaigangan Biodiversity webinar series. I'm Lailani Masungsong and I will be your host. So before we start, kamusta po ang lahat? Nakapag-breakfast na ba? Ayan. We would like to know more about you. So can you please chat your name and your affiliation in our chat box so we can acknowledge you po. So I see familiar faces po kasi. Ayan. I see welcome po sa ating webinar, Dr. Nonatus S. Bautista from the Plant Biology Division, IBS. Ayan, sino pa po ba? Kindly state your name po para po makilala natin ang isa't isa. Ayan, I see, sino pa ba dito? Ayun, welcome po and good morning, Dr. Maria Celeste Baneticla Hilario, our project leader for CONserve-KAIGANGAN. And sino pa po bang nandito? Parang shy po ang ating mga participants. Walang nag-chat sa chat box. Ayun, welcome din po, Sir Giro Adorador from the Plant Biology Division. Ayan, may nakita po ako. Welcome po, Ma'am Menisa Aquino. Antonio from MMSU. Ayan, Ma'am Lisel Magtoto from UP Baguio. Welcome po sa ating Kaigangan webinar series. Ayan, Sir Marlon Rivera. Welcome po from Japan. Ayan, from the Plant Biology Division din po. Ayan, at sino po ba? Ma'am Rod Delina Deito. Ayan, good morning po. Good morning everyone. Ayan. So salamat po sa pag-attend nitong aming webinar. So again, welcome to our CONserve-KAIGANGAN webinar series. So this webinar happens once or twice a month. So stay tuned po in our FB page. Ayan, meron pa po. Hello, good morning, Ma'am Caroline Arbotante from Angeles University Foundation. Good morning, ma'am. Ayan po. So, I know all of us are excited, but before we begin, we want to remind everyone that this webinar is recorded. So with this, please take note of the following rules for our decorum. So first, kindly keep your microphones muted, especially when our speaker is presenting. Second, if you have questions or clarifications during the presentation, please send your questions in our chat box and we will acknowledge that during the open forum. So the link to the evaluation form will be provided and e-certificates will be sent after completing the evaluation form. So make sure lang po the email address you will indicate is valid and correct, or it will be the basis for sending the e-certificates. And now that we have set our ground rules, we can now continue with our event. So before we call our guest speaker, let me first introduce her. So our guest speaker is an assistant professor from the Plant Biology Division, Institute of Biological Sciences, University of the Philippines, Los Baños. So she teaches basic botany, plant physiology, and economic botany. Her research interests are in plant natural products. So without further ado, let us all welcome Assistant Professor Elea Villaverde to talk about metabolite screening of two important native plants in Kaigangan Forest through high-performance thin layer chromatography.
Hello, good day. I am Eleanor C. Villaverde, and on behalf of my colleagues, Assistant Professor Medina, Assistant Professor Rivera, and Sir Junel Colobong, I am here to present a portion of our findings from Project 2, entitled Ethnobotanical Survey, Usage Validation, and Safety Assessment of Economically Important Native Plants in Forest over Limestone Ecosystem. It's entitled Metabolite Screening of Two Important Native Plants in Kaigangan Forest through High-Performance Thin Layer Chromatography. So our group was part of the first phase of the Kaigangan project, and this is a short talk that is based off of some of the results that we obtained from the project. Moving on, it's important to note that plants are widely used in our lives. So we use them as food, we use them as perfumes, for dyes, poisons, and as pesticides, but more importantly, as medicines. Okay, so pictured here in the diagram are the different properties or the different effects that we get from plants when we use them as medicines. Some examples of the plants include lemons, pepper, rosemary, and oregano. Okay, and in order for the plants to be able to do those properties, these ones are entirely dependent on their structure. Okay, so because we know that in science there is a correlation, or there is a clear connection between the function of the substance and its structure. As pictured here, we can see that there are substances that have a lot of OH groups, and some substances here have charges, while other substances are predominantly hydrocarbon in nature. And these differences, having OH groups, having charged units, usually are characteristics of polar substances, while those that only have hydrocarbons are more of nonpolar substances. Okay? I have distinguished or given characteristics of polar and nonpolar substances because it is one of the ways that we can differentiate the components in a plant extract. Because when we extract the plant, when we juice the roots, the stems, or the leaves, or the fruit, it's not just one component that is present. So there are a lot of components present, and they may be both polar and nonpolar. So their solubility is something that we can use in order to differentiate and to separate both. And that is a core principle in chromatography, wherein you have a stationary and a mobile phase. The stationary phase is where you place your extracts, as pictured here in our diagram. It's the rectangular illustration, and the extracts, we can think of them as the pink dots. And the mobile phases usually are solvent systems, which are depicted here as your teal liquid inside the container. Okay, so we can see here that the stationary phase, or the compounds in the plate will be and the solvent phase will run through the stationary phase, and in the process of capillary action, we can see here that there are two kinds of dots in our illustration, the red and the blue ones, that will be produced from the original pink extract. So this means that we are separating whatever is in the pink extract by virtue of its polarity, or by virtue of its solubility with the mobile phase and with their attraction with the stationary phase. Okay, so this is an example, or this is a generalized example of thin layer chromatography, wherein the thin part is attributed to the stationary plate, and the other concept here pictured is the RF value, which would be the characteristic value of the spots once the process is completed, because the RF values, or the retention factor values, are obtained as a ratio of the distance traveled by your samples, the blue and the red lines, over the distance traveled by the solvent, which is symbolized by the teal line. It's number 4. So with that, we can determine the identity of a substance depending on the solvents, or depending on the factors that we used in order to separate them from the mixture. Because if ever we used a different kind of solvent, let's say we used a nonpolar solvent, maybe in our illustration, the spots will be interchanged, that the blue spots will travel further because they may be more nonpolar, and the red spots will travel lesser because they are not attracted, or they are not matched, or they are not friends. You can think of it as they aren't friends with the teal solvent. Okay, so far we've just discussed about plants and their secondary metabolites, the solubilities, the principle of solubility, and a little bit of background about chromatography. So I mentioned in our title that we used a certain kind of chromatography, the high-performance thin layer chromatography. And you can see here that for our study, we targeted 3 groups of secondary metabolites. We looked into the flavonoid, essential oil, and alkaloid profiles of the 2 plants that piqued our curiosity during our survey in Samar. You can see here in the diagram that there is something called as a CAMAG, which is actually the setup for the HPTLC, and that there are different equipments centered throughout this illustration. And these different equipments play certain roles, or they have different functions in carrying out the high-performance thin layer chromatography analysis. And you can see that they are all connected, or central to the illustration is the vision cats, which, yes, the icon looks like a cat, because the vision cats is the software that controls the different equipment. So let me explain further. So the HPTLC setup, as I mentioned earlier, it's a plug-and-play setup wherein you have your vision cats as the software that sets the controls for all of the equipment that you see here in this illustration. So originally, thin layer chromatography is done manually. You use capillary tubes or toothpicks to obtain your sample and to make a small prick or a small dot on the TLC plate that is very centered and precise on the starting line of the TLC plate, and then you will and as you saw in the illustration, the diagram earlier, you just place it in a container and let capillary action flow, let the solvent flow through capillary action, and then you will obtain the plate, and then you will mark the different spots. For HPTLC, that's still the same thing that you are going to do generally, but you have now the different equipments that make this process semi-automated, wherein you have a machine that will dispense the extracts uniformly and in a more controlled manner, and even in smaller doses than what you were manually dispensing through the Linomat 5 machine. And then afterwards, you will be placing the plate inside the development chamber or the automatic developing chamber, wherein it will take care of the saturation or the setting up of the chamber, and it will take care of the drying of the plate after the solvent has run through the plate. And afterwards, for our setup, we didn't have a automated derivatizing machine. We still made use of the glass sprayer, but still, afterwards, we were able to document or to take a very nice picture of the plates under different conditions, under white light, under different nanometers of UV, short UV, 254 nanometers, and long UV, 366 nanometers. And we were able to observe the different colors and the different behaviors of the bands depending on what secondary metabolite group they were. And the last equipment, which is the TLC Scanner 4, it's for evaluation. So this equipment is responsible for the quantitative part of HPTLC, especially if you already have the standards, the reference standards, the gold reference standards, wherein if you run it side by side with your sample, after scanning it, this machine can be able to give you values of how much of the extract or how much of the target compound is in the extract, since you have a standard, since you have run a standard with your sample. So it can compare, and it can make a calibration curve, and then give you an idea of how much of that target compound is in your sample. So these succeeding slides are just a closer look with the equipment. So again, so for the Linomat 5, this is the semi-automatic dispenser of the extract. So you can have around 20, we use 20 UL, we put it in the syringe, and then it dispenses the samples in a band manner, so it's more orderly. And then afterwards, we place it inside the developing chamber, and wherein it will be the conditions are already controlled, the humidity, and then the conditioning of the plate, of the chamber, and then the drying of the plate in order for it to be viewed in the visualizer afterwards, wherein, again, as I mentioned earlier, you can view it under white light, under short UV, which is 254 nanometers, and under long UV, which is 366 nanometers. And in association with the VisionCat software, you can place labels on the picture of the chromatogram once you have taken a photo of it, and you can also have you can also place the RF values by clicking the bands. And as I mentioned earlier, the TLC scanner 4. This one is responsible for giving quantitative values of the extracts, especially if you have run a gold standard or a reference compound along with your sample. So for our study though, and for this talk, I will only be showing up until the product of the TLC visualizer. So we for the quantification, we were still figuring out this process. Okay, so this is just to present the software associated with all of the hardware. We're in operating the equipment. So the commands are channeled through the VisionCats software. And this is a picture of the home screen, and you can actually click the icons in the order that you plan to use them. So usually for our analysis and training, we start off with the visualizer first. We like to take a look at the plates as they are, because we want to ensure that before we do place extracts on them, that they are debris free, or if they do look okay with our naked eye and under the different kinds of light, we then detect some debris or a malformation. Then it saves us the effort of only finding out about it if it will, and most probably it will interfere with downstream processes if we don't detect that the plate initially is not up to a good standard. So after knowing or after seeing that the plate is usable, then we click on the Linomat 5, and then we set the parameters. We set that all samples must be dispensed for 20 UL in a certain in a band form, and then we click the automatic development chamber, wherein we will be subjecting the plates to the solvent system afterwards for development. And then if ever we do need to enhance the visualization or the visual display of certain secondary metabolite groups, then we select the derivatization spray, although again, we just use a manual glass spray for that. But we do visualize it after derivatization with the visualizer to check if there are differences with the initial appearance versus the ones then when we apply the visualizing agents. And then we tried to obtain information about its amount through the scanner. But again, we were at most successful with the visualizer equipment. So here are examples of chromatograms under white light, 254 nanometers and 366 nanometers. And you can see that there are variations in their color. For white light, there are a lot of bands that are not visible when viewed only with that illumination compared to 366 and 254. So for 254, these are the bands that when hit with shortwave UV, the darker bands or the bands that appear dark at this stage tend to undergo quenching, fluorescence quenching, and those bands can also be seen at 366 nanometers when given long-range UV light, but they manifest, but the other bands are now more visible at this stage. So again, this helps in qualitatively detecting the different components that are present in your extract that may not be visible with the naked eye. So to compare the TLC and the HPTLC, so here's just a short rundown of the advantage of HPTLC and its disadvantages. For advantages, as I've mentioned earlier, it's a big help that it's automated, because if you have shaky hands or something goes haywire during the process, so the errors are minimized and the plates or the band development is as much as possible uniform and can be easily read, and the extracts separated, the extra components separated well. Resolution is also enhanced since, as we have seen in the slide earlier, so we can see bands at different, we can see bands that are not visible at visible light under different UV wavelengths. And at the same time, you have HPTLC is also able to separate the components of an extract, even though some components are only in little amounts in that mixture. It's still able to see it. It's still able to differentiate it from the others. Okay? And with the equipment present in HPTLC, documentation is also made easier. And if ever you also have the standard, then you can further elevate the analysis by quantifying how much of that component is in your extract. For the disadvantages, though, HPTLC involves a lot of specialized equipment, as you have seen earlier, and that in order to operate this well and to maximize the use of each, additional training is needed for the people that will be using it in the future. And you also have a limited plate length. So if ever you need a longer plate to separate the identity of the mixtures, then it will be more difficult in HPTLC due to that limitation. And finally, the specialized equipment does cost a bit much, as well as if you will also be buying the standards. But still, you can make use of HPTLC for qualitative purposes if ever cost is still a problem for the quantification part. So that's a bit of a hefty introduction with HPTLC. So now let's focus on the plants that we obtained from Samar. So Samar is a curious place because its soil still has limestone. So as you have read in our title, we obtained it from Kaigangan, which is the characteristic of Samar soil. It still has limestone in it. So we know that plants are dependent on their environment, and it's a curious case for the plants to be growing in soils with limestone. So we obtained, we surveyed Paranas, Taft, Basay, and Giwan and we asked elders and those that are more knowledgeable with the different plant uses about the plants that they use for medicines, for food, for textile, and other purposes. And after tallying the results, so we have Arcangelisia flava and Spondias pinnata as two of the many plants that piqued our interest. So you can see here that Arcangelisia really has a yellow stem, and then Spondias is a small, and this picture is still a small shrub that is found in the backyard of one of our respondents. So a little bit about Arcangelisia flava that belongs to family Menispermaceae. Its common name is albutra, and here are its traditional uses: for wounds, for skin irritations, as a stomach kick, for fever. It's an expectorant. It helps with periods, or it can also induce abortion. And for pharmacological studies, it is found that it has antifungal, antibacterial, anti-tumor, antiviral properties, And it could be due to the high amount of berberine that is found in its stem. So pictured here as well, the very yellow stem of albutra. Spondias pinnata, which is commonly known as libas, is described as a souring agent in a variety of dishes and soups for Samar. And we have a general idea that if something is used for this purpose, most likely it has a lot of vitamin C. Okay, and apart from that, it's also documented to have wound healing, anti-diabetic, antioxidant, diuretic, antimicrobial, and anti-inflammatory properties. So when we obtained the stems of Arcangelisia flava and the leaves of Spondias pinnata, and then we extracted them with methanol and prepared them prepared 3 sets of extracts, because one extract will be subjected to a flavonoid analysis, the other to an alkaloid profile, and the other to an essential oil profile. So each secondary metabolite group has their own preparations and different solvent systems in order to separate the target components from the mixture. So for albutra, here are its flavonoid, alkaloid, and essential oil profile. You can see for flavonoids, it has a blue band at the bottom and then a yellow-green band at the top. And then for alkaloids, you have here an orange-brown band, an extensive band. And then for essential oils, compared to the two other ones, it seems as it has lesser content of essential oils compared to the flavonoid and alkaloid group. For the flavonoids, usually those that appear in blue bands are of chlorogenic acid or phenol carboxylic acid identities. And then usually at this position, it's around the identity of quercetin. For alkaloids, usually this is for the identity of berberin. For Spondias pinnata, or for libas, you can see it has a different flavonoid profile compared to your albutra, and as well as for the alkaloids. And if you will look at its essential oils compared earlier, it has a more diverse profile compared to your albutra. So again, it has differing appearance of bands at different positions and of different colors, which gives you an idea that they have different components inside of them. So so the bands here pictured at the other side of the slide. So the first band is, if you remember, is that of albutra, and then the second is of libas. So they have entirely different colors of the bands present. And for flavonoids, we use the citrus peel, gallic acid, and quercetin as standards, and developed using this solvent system. And derivatization, that means we intensify the bands after development by applying the natural products and polyethylene glycol reagent. And again, it did intensify the fluorescence, as evidenced by the yellow and the orange bands. So at 366 nanometers, that's the wavelength best to observe the flavonoid profiles. You can see here that the yellow-orange, the yellow-green can be the camphorol or the a flavone or a flavonone, a flavanol identity. So for the yellow, for the orange, it may be of a flavone identity of luteolin. So, and for the blue fluorescence, it can be phenol carboxylic acids. So we based these putative identities according to the TLC Atlas of Wagner and Blatt, 1996. So We used the same solvent system that they used, and we tried to stay faithful to the conditions for flavonoid development. So for flavonoids, we are aware, or if you've heard of them, that they are ubiquitous in nature and they can be found in fruits, in leaves, in grains, and in other parts of the plant. And they are usually associated as antioxidants due to their structure. So their structure that enables them to act as anti-cancer, anti-inflammatory, antibacterial, and all of that is usually attributed to their structure, the presence of their double bonds. So camphorol is also another kind of a flavonoid, and that is also, and its antioxidant capacity is also attributed to its double bond structure. For the alkaloids, pictured here is the first band of the albutra, and then the second is of libas. We use the piper seeds and berberine and this solvent system, and the visualizing agent is the Dragendorf's reagent at visible light. And again, you can see with this chromatogram that it looks different from the 366 nanometers of the flavonoids. So for here, it's at visible light, and the albutra sample tested positive for alkaloids. And most likely, this is the identity of berberine. So berberine is an alkaloid that confers several health benefits, similar to the flavonoid. So one of its main one of the main features of its structure that can enable it to act as an antioxidant and to facilitate and to improve cardiovascular health and other effects is due to its double bonded structure. For essential oils, as you can see here, again, the first picture is of your albutra, and then the second is of your libas. And the standards that we used were rosemary and oregano leaves. Given this solvent system and this visualizing agent, so there are a lot of bands that showed up for libas compared to your albutra. And essential oils, as I mentioned earlier, these belong to the terpene group, wherein they have a lot of hydrocarbons. Okay. And usually these hydrocarbon structures are volatiles and aromatics. So if you have eaten rosemary chicken or you have ingested oregano for your cough, so you are familiar that they are more fragrant compared to other plants. So they have a certain fragrance that is emitted from their leaves due to the volatile oils present. So the essential oils have a lot of roles in our day-to-day life, and they can also confer health promoting characteristics such as anti-cancer, antimicrobial, anti-inflammatory, anti-allergic, and as insecticidal due to their antioxidant properties as well. Okay, so this slide just presents side by side the different chromatograms of the different secondary metabolites of your albutra located at the left and your libas located at the right. So as you can see here, they have different compositions of flavonoids, of alkaloids, and of essential oils. And these lend, and again, as I mentioned earlier, their components lend to the activities or suggest why they can perform those certain activities when taken in by humans. Okay, with regards to additional studies or the latest studies regarding albutra, so it was included as the leaves were actually studied by other researchers, and that they charted the different phytochemical components of albutra leaves. But for these three studies, these are related studies wherein they examined the stem also of albutra, but it's from Quezon. And then they did a differential extraction wherein they subjected the stems to different solvents. And as we have learned earlier, or I hope you learned earlier from my discussion, if you use a different solvent, there is a possibility that you will be getting a little bit of a different profile compared when you used another solvent because of the polarities of the substances inside the plant extract. Okay? And with regards to berberine, so berberine was in this study was also a candidate for to help out with during the peak of the pandemic of for COVID, to alleviate COVID-19 symptoms. So for libas, for the latest studies about libas, it's more of its hypoglycemic potential, of how it can also generate as a tree from its seed. And very interesting that its culinary application has been explored. And if I remember my research right, or when I browse the literature, I know that it has already been, or another state university, PUP if I'm not mistaken, has already tried to formulate a sinigang mix that, or an additional condiment that contains libas leaves. So which is quite impressive and quite entertaining, because our group first heard of libas from Samar as a souring agent, and then it's nice to see that the recent research done by other researchers has also expanded further on this property. So with that, here are some let's review some of the main points that we discussed for today. Wherein Samar Island has an abundance of plants used for various uses, and two of those that we obtained from our previous study is albutra and libas, and they are rich in secondary metabolites. And we took a look at their flavonoid, alkaloid, and essential oil profiles through HPTLC, and saw that they were different, and these differences may lend to their activities and uses. And it is recommended that additional screening of plant secondary metabolites should be done, and it is possible to do it in a more automated way when HPTLC is used. And different parameters can also be implemented in order to further add to the research about these substances. With that, here are my references, and I am now open for your questions. Thank you for listening.
Thank you very much, Ma'am Elea. Very interesting and wonderful talk. So I know our audience or participants have a lot of questions in mind. So just a reminder po, for this open forum, please type your questions in our chat box and we will read them for you. So, Ma'am Elea, can you open your campo? Hi, ma'am. Good morning.
Hello po. Good morning
Ayan.
to everyone.
Again, ma'am, thank you very much for accepting our invitation. So while our participants are typing their questions, I have here some questions from our pre-registration. So for the first question, ma'am, Sabi dito, were there any unexpected findings or challenges encountered during the metabolite screening process?
Um, for the metabolite screening process, so the main difficulty that we encountered as a team was that this analysis was done during the peak of the pandemic. So mobility was limited, okay, for us to go to the lab and to analyze it. And if I remember correctly, it was our project leader, Sir Marlon, and our project staff, Mr. Colobong, who did most of this analysis because they were near to the lab, as I and my other colleague were situated in our homes. But for the results per se, when we took a look at the literature, there wasn't any unexpected ones. It's more of just that we were fascinated that they had a lot of, as you can see in the HPTLC plates, they had a lot of bands. Although we just generally knew that they contained a lot of essential oils, we were just really fascinated that there was a lot of bands or contents more visible compared to the usual TLC process.
Ayan. Thank you very much, ma'am. For another question, since, ayan, sandali lang. So, for the second question, ma'am, how might the findings from this study influence future research for conservation efforts in the Kaigangan Forest?
All right, thank you for that. So for since at this level you can at HPTLC level you can qualitatively profile whatever they have inside of the plants, and once combined with the current or existing literature that explains their functions, then at least you have an idea of their content, and you can further advocate for them to be monitored or to be observed more closely, or you can also get samples from SAMAR and try to compare it with other areas, or with other areas that also have that same plant, in order to see or to compare if their profiles, if they are used for the same thing, but if their HPTLC profiles or their contents are quite different, then at least that's a point to look into, to consider if their location can also affect the contents of of the plant extracts. So for conservation, then if you know how to use it, why you're using it, and what are the essential components that make it possible to be used for that purpose, then it adds to the value of the plant being conserved. Although generally plants do need to be conserved, but at least there is an additional reason to back it up as to why to make sure that they won't disappear even before we get to know them better.
And thank you very much, ma'am. So we have here sa chat box, hello po, from Mr. Melvin Castro Santo. Hello po, may pre-suggested mobile face po ba ang instrument, or need pa din mag-try to optimize?
Okay, hi Sir Melvin. Ayan, so for our procedure, we opted to observe the recommended procedures by Wagner and Blatt sa kanyang TLC analysis. So we just followed the proportions, and then we went with the solvent system that is documented. The optimization comes with how much plant extract should be loaded onto the syringe or to be dispensed, because sometimes, although we try to have a ballpark estimate na 10 UL lang, sometimes the bands seem to be too thin, or if we try to increase the amount, sometimes the bands then tend to spread out. Therefore, hindi na malinaw kung saan saan maiging kunin yung amounts para maka-create ng RF value. So solvent system wise, we just follow what is documented, since medyo constant na naman siya. More of the procedures to carry it out, yun yung may optimization.
Ayan, sabi ni Sir ay thank you very much, ma'am. So, meron pa ma'am from Mr. Daryl Seribo. So, good morning, ma'am. May I clarify if you use different wavelengths in observing different types of phytochemicals such as flavonoids, alkaloids, and essential oils?
Hi, Sir Daryl and the rest of CBGRL. We did use different wavelengths. So with each result, we typically observe them using the 3 wavelengths. We observe them at white light, and then depending on what is stated from the TLC atlas ni Wagner and Blatt, we try to double check if it does need application of intensifying agents for visualization. If it does, such as in the case of flavonoids, we initially view it, we initially view it with white light, 254 and 366. So, and then if the literature says na, oh, it needs intensifying reagents to further visualize it, then we apply those reagents, and then we view it again using 366 and 254 for comparison. So it's largely dependent on the secondary metabolite group that you are targeting if it needs certain changes in the wavelength that you will be observing. But again, for HPTLC, it's best to view it using the 3 illuminations, just so to check if there are certain differences, or maybe although the literature says that there isn't anything to observe at different wavelengths, and it's still double checking that you might find something new or something different from what is documented.
And thank you very much, ma'am. So, due to time constraints, medyo marami-rami pa po yung questions, but I will read another one, ma'am. And for the other questions, you can ask directly, Ma'am Elea. We will provide her email address so you can ask questions for her. So, another question, ma'am, from Ma'am Menisa Antonio. With your promising results on these species, ma'am, what are your next plans or ways forward? For example, elucidation or product development.
Hi, Ma'am Melissa. Thank you for your question. As of now, we haven't gone back yet to Samar as a team, as the original team, to further obtain more samples. But one thing that we were initially planning to do, but we were hindered due to the initial part of the pandemic, is that to hopefully obtain, like for the case of albutra, to obtain different samples of albutra from the different parts of Samar, because we would like to check if there were any variations. Although they are all growing in Kaigangan soil, there might be certain variations with the different parts of from being obtained from different parts of Samar. Although for the product level type of research, we are not yet at that area, because the crude extracts still contain a lot of other phytochemicals, and that needs more sophisticated equipment in order to isolate or to pinpoint just one phytochemical to try to process it to the next type of product. For Spondias or for libas, as I mentioned earlier, I'm quite surprised and amazed that another university has already tapped into commercializing the libas as a souring agent. So I'm just not sure if it's already released in the market, but I read the findings and their application for patent in the literature over at the internet.
And thank you very much. So with that, thank you very much po again, Ma'am Elea, for that very engaging and lively discussion. Okay, so it looks like we covered naman po all the questions. So yung mga hindi po nabasang questions, again, we will provide Ma'am Villaverde's email address, so you can ask her na lang po personally. So, we'd like to present the certificate of appreciation for Ma'am Elea. So let me just read the citation. So, University of the Philippines, Los Baños award the Certificate of Appreciation to Ma'am Eleanor C. Villaverde for serving as the resource speaker during the webinar presentation entitled Metabolite Screening of Two Important Native Plants in Kaigangan Forest through High-Performance Thin Layer Chromatography of the Kaigangan Biodiversity Webinar Series held on 24th June 2024 via Zoom. So, signed, Inocencio E. Buot Jr., Professor and Project Leader. Ayan. Again, thank you very much, Ma'am Elia. Can we give Ma'am Elia a big round of applause by sending a heart reacts? Thank you, ma'am. Thank you, thank you. Thank you very much. And for the next segment, so for the next episode, we have here on July 29, 9 a.m., again, by Zoom, we have Forester Marne G. Origenes that will discuss the mapping the distribution of top priority plants in the forest over limestone landscapes of Summer Island Natural Park, Philippines. Ayan. See you again po on the next webinar episode. So before you leave, please do not forget to scan the QR code or visit the link shown, or the evaluation form will the evaluation link will be posted also in our chat box. So thank you very much again po, and enjoy the rest of the day.
Maraming salamat po ulit. Ingat po
Thank you
lahat.
very much. Ingat po.
Summer Island is the third largest island in the Philippines and has one of the most extensive forest over limestone ecosystems in the country. This ecosystem is home to a wide array of uniquely adapted flora and fauna. Summer Island is known to be one of the centers of botanical diversity in the Philippines and has been nominated as a UNESCO World Natural Heritage Site for its exceptional natural beauty and precious biodiversity. For these reasons, CONserve-KAIGANGAN, a biodiversity research program, was established. Kaigangan is a Samar local word that means forest over limestone. Our research program's mission is to assess and conserve the biodiversity in Samar Island's forest over limestone towards its sustainable management. One of our program's projects is the assessment of biodiversity and community structure in forest over limestone ecosystems. This component of our research aims to determine plant diversity and vegetation structure in various habitat types in forests over limestone, to determine animal diversity associated with forest over limestone, to identify functional classification of vegetation with its associated animal diversity, and to analyze the environmental factors influencing the prevailing structure. Determine animal diversity associated with forest over limestone, to identify functional classification of vegetation with its associated animal diversity, and to analyze the environmental factors influencing the prevailing structure of the biota. We conducted a comprehensive biodiversity assessment of forests over limestone in the municipalities of Paranas, Taft, and Giwan with remarkable findings. A combined total of 71 plant species were recorded from these municipalities, with at least 3 tree species classified as vulnerable based on the DENR updated national list of threatened Philippine plants. A total of 52 terrestrial vertebrate animal species were also recorded, including 5 amphibian species, 15 reptile species, 20 bird species, and 12 mammal species, with conservation statuses ranging from threatened to endangered and critically endangered. based on the DENR updated National List of Threatened Philippine Fauna. This knowledge will empower our stakeholders in these areas to make appropriate research-based conservation efforts for the protection of these ecosystems that harbor an assemblage of rare, endemic, and economically important flora and fauna. Through this program, people will be equipped with knowledge on forests over limestone biodiversity, including the ecosystem services that it can offer to the community. We at CONserve-KAIGANGAN continue to further raise awareness and capacitate national and local government agencies, especially local communities living in and around Samar's Kaigangan.